Cell membranes are structures of contradictions. These oily films are hundreds of times thinner than a strand of spider silk, yet strong enough to protect the delicate contents of life. The cell's watery cytoplasm, genetic material, organelles, and all the molecules it needs to survive. How does the membrane work, and where does that strength come from? First of all, it's tempting to think of a cell membrane like the tight skin of a balloon, but it's actually something much more complex. In reality, it's constantly in flux, shifting components back and forth to help the cell take in food, remove waste, let specific molecules in and out, communicate with other cells, gather information about the environment, and repair itself. The cell membrane gets this resilience, flexibility, and functionality by combining a variety of floating components in what biologists call a fluid mosaic. The primary component of the fluid mosaic is a simple molecule called a phospholipid. A phospholipid has a polar, electrically charged head, which attracts water, and a nonpolar tail, which repels it. They pair up tail to tail in a 2-layer sheet just 5 to 10 nanometers thick that extends all around the cell. The heads point in towards the cytoplasm and out towards the watery fluid external to the cell, with the lipid tails sandwiched in between. This bilayer, which at body temperature has the consistency of vegetable oil, is studded with other types of molecules, including proteins, carbohydrates, and cholesterol. Cholesterol keeps the membrane at the right fluidity. It also helps regulate communication between cells. Sometimes cells talk to each other by releasing and capturing chemicals and proteins. The release of proteins is easy, but the capture of them is more complicated. That happens through a process called endocytosis, in which sections of the membrane engulf substances and transport them into the cell as vesicles. Once the contents have been released, the vesicles are recycled and returned to the cell membrane. The most complex components of the fluid mosaic are proteins. One of their key jobs is to make sure that the right molecules get in and out of the cell. Nonpolar molecules like oxygen, carbon dioxide, and certain vitamins can cross the phospholipid bilayer easily. But polar and charged molecules can't make it through the fatty inner layer. Transmembrane proteins stretch across the bilayer to create channels that allow specific molecules through, like sodium and potassium ions. Peripheral proteins floating in the inner face of the bilayer help anchor the membrane to the cell's interior scaffolding. Other proteins in cell membranes can help fuse two different bilayers. That can work to our benefit, like when a sperm fertilizes an egg, but also harm us, as it does when a virus enters a cell. And some proteins move within the fluid mosaic, coming together to form complexes that carry out specific jobs. For instance, one complex might activate cells in our immune system, then move apart when the job is done. Cell membranes are also the site of an ongoing war between us and all the things that want to infect us. In fact, some of the most toxic substances we know of are membrane-breaching proteins made by infectious bacteria. These pore-forming toxins poke giant holes in our cell membranes, causing a cell's contents to leak out.
Hey guys, so I started off with that TED Ed video. It's a really nice synopsis, though some of their stuff is a little weird looking, like the way their arrows go, but it's a really good synopsis to get us started talking about membranes today. So we're going to be talking membrane structure and some transport, a lot of review, but I'm hopefully to just really get us thinking about cell membranes and where they are inside the cell. So we're not talking just the outside of the cell, but just all the internal membranes as well, which we talked about organelles last week. And so let's just get into it. So the last thing they talked about before we get too deep, cytolysins is what they were talking about. So pore-forming toxins, or PFTs. There's a variety of PFTs, CDCs, cholesterol-dependent cytolysins, is one subclass of those, and there's a bunch of subclasses of these. Lots of gram-positive bacteria have CDCs. They're actually thiol dependent, which that's like sulfur hydrogen, like disulfide bonds. But they're cholesterol dependent because there's so much cholesterol in cell membranes, and we're going to talk about that a little bit. But you can see they form pores. Another example of this would be like perforin that gets secreted by your immune cells to kill bacteria. And so the idea of having a pore-forming protein structure that like pokes holes and stuff isn't that crazy. We see that independently evolve over basically all forms of life. So that's really cool. Now, let's get started talking about not envelopes, membranes. And membranes are everywhere, obviously. I found this, so I'm going to show a few pictures of these from Draw It to Know It. Awesome. I love Draw It to Know It. They make really good pictures. They're like really great, just synopsis of stuff. But almost everything you can see on this picture has a phospholipid bilayer. And phospholipid bilayers are what we're going to talk about today. So that scaffolding that is, we usually think about it on the outside of the cell, right? But it doesn't necessarily have to be. It's all over the cell. I mean, every single one of these organelles has a phospholipid bilayer, sometimes two, like with the nuclear membrane or with the mitochondria or like a chloroplast. And so it's really important that we understand how they work, because without phospholipid bilayers, they don't work, cells. And so we need to have it be semi-permeable. That's the most important part, right? So we can have gas exchange. In general, we can have water exchange. But for the most part, most things, you require a protein to allow them to enter or exit. They're extremely dynamic processes. They're changing all the time. The fluid mosaic model is the most current model, though I have some review papers if you'd like to see them about, there are some modifications to the physics of how that actually works. But it's the scaffolding as well. So not just do we let things in, do we let things out, but that structure isn't just the physical structure of the membrane. It's the attachments to it. And so we have our internal attachments, so our cytoskeleton. They have to be anchored somewhere, right? And so they're going to be anchored to that membrane. And then you also have the extracellular matrix, because we're not usually talking about individual, like single-celled eukaryotes in this class. We're usually talking about multicellular, like us. And so we have like tons of different cells inside our bodies, and there's lots of extracellular matrix, and so we need to have ways of those things being connected, right? And so that's where our cell membrane comes in. We need to talk about what membranes are made of. Now, this comes from a review paper. I have a couple of review papers that I grabbed stuff from. So we need to talk about what our membranes are actually constructed out of. And in general, it's some kind of charged head, so a hydrophilic head, and then a hydrophobic tail, okay? But we can just change, we can put different things in those spots, and then that changes their name. But overall, their structure is the same. And so you end up with, like, you can have, it just depends on what it is. So like if you change the head, you can change like what's on the top. Like it can have a phosphate in one portion, and then you stick other stuff. You can stick a sugar on there. You can stick proteins on there. If you start changing what the chains are, so the lipid chains, so usually they're unsaturated fatty acids, but it kind of depends. So you can have your saturated versus unsaturated. Remember that in natural biological systems, your unsaturated fatty acids are cis as opposed to trans, hence, trans fats. And so we get nice bends in these. A trans fat would look straight, whereas our cis fat tends to bend. And so that gives us a little more flexibility, and it allows your membranes to move a little bit more. But if you start changing the length of these, which they don't usually vary too much, maybe between 18 to 22 carbons in length. But you can change, like maybe you don't want two identical unsaturated fatty acids next to each other for your two little legs that are sticking out. So you swap one out for a different one. And then you get kind of like crooked looking ones like this. There's so many possibilities. Maybe you want a sphingolipid. So one of them is sphingosine as opposed to a fatty acid. You can just change everything. The other thing that you can see on these charts that gets thrown around is cholesterol. And the presence, at least in animals, is cholesterol. Okay, that can vary based on what kind of creature you are. So if you're not a mammal or an animal, then like funguses have ergosterols. Okay, so you've got ergosterol down here. Plants have phytosterols. They don't look that different. They have similar structure, but they are different enough that they have different names. And so generally that's a way to tell creatures apart too, is by what kind of sterols they actually have in their cell membranes. Now, sterols are really important. The amount of sterol, on average, it's actually a 1-to-1 ratio, phospholipid to cholesterol in your membranes. Cholesterol, we've talked about this in biochemistry, if you've taken biochemistry. The presence of cholesterol in your cell membranes, it almost acts as a buffer. So if your cell membrane gets too cold, it keeps it nice and fluid. And if it gets too hot, it also it can kind of like stick it together so it doesn't fall apart. And so that's the function of having sterols in your membranes. They maintain fluidity regardless of temperature is generally the main function. Another important part, I mean, you've got those charged heads, and I mentioned a second ago that you can add different things to them. And oftentimes we usually think of not adding proteins so much, because usually we will embed proteins like as large protein structures. You can tack amino acids on there, of course, but for the most part, we usually think about throwing sugars on there. So like glycosylating your phospholipids. And that's how we will add on, sugars for like oligosaccharides for communication between cells. How do your cells actually know what the other cell that it's seeing is? That's a good question. How do cells tell each other apart? Well, they generally put different sugars on their surface, and that's how they talk, which is kind of fun. So jumping off of that, just this is a drastic oversimplification, of course. You can dig real deep into the literature about the actual membrane contents and the stuff that can go into cells. So if that kind of tickles your fancy, awesome. I have some papers that you can read. Now, if we keep on going, it kind of makes sense. Imagine if you had all of your phospholipids had really long tails and lots of unsaturated fatty acids. They would kind of spread out more, right? Because their little legs are kind of bent, like their knees are bent a lot. As opposed to very dense saturated fats, that's going to change how tightly you can pack them. And it turns out there's a lot of biophysics involved in understanding how you can actually change membranes, change their curvature, and thus change how they interact with each other and how they serve in their different functionalities. And keep in mind, this isn't just the cell surface. This is all of your endoplasmic reticulum, your Golgi, your nuclear membrane. All of those membranes can have different amounts of different lipids, and it's crazy. Like when you start thinking about that, like maybe your ER has like maybe the trans Golgi has different contents. Maybe the cis Golgi has different phospholipids. Oh man, it just gets super complicated. And I'm just showing this to you. This comes from a really nice Nature Reviews molecular cell biology paper, and it's talking about the biophysics involved of creating different membrane curvatures, and they go into hardcore depth. So I'm way out of my element talking about this in general, but just there's some cool stuff going on. Being able to embed things and have them move around in the membrane is really the most important part, as well as having your membranes be able to move. Now that depends. So if you're like a macrophage and you need to crawl around through tissues, awesome. You're probably going to have, more malleable membranes, as opposed to like an epithelial cell. You don't really want your membranes to change that much. And so that's going to alter what your contents of your membranes actually are, which is kind of cool. There's also an extensive amount of research that I'm not going to go into that talks about how you actually build your membranes. They're complicated processes. Could I gloss over it any more than that? But basically, you have proteins and things that are associated with, building your phospholipid bilayers. And destroying them as well, because things go bad. You need to change things up based on all kinds of stuff, and so there's a lot involved in that. I recommend going to this paper if you want to read more about it. But yeah, it's really, really interesting if you're super into membranes, which some people are, and maybe you are. So jumping off of the idea that membranes are complex and full of all kinds of crazy stuff, we can now talk about something that's a little more fun, which is how you actually stick proteins in there. So we know that when we send our DNA, we make our RNA off of our DNA, and we send that RNA out to a ribosome. It gets made. It will have a special signal on it that will tell it to bind to the endoplasmic reticulum, and it will also tell it whether or not to have that protein, the amino acid chain, be embedded within the endoplasmic reticulum, or to be just kind of secreted into the lumen of the ER and then can be like secreted out like through a vesicle. Now, we're generally talking more about embedding our proteins. And there's lots of different embedding proteins, or embedded proteins. We call those transmembrane, though they don't have to necessarily traverse the entire length of your membrane, like both phospholipids layers. So you can have that be just monolayer associated. So like this one here, we can have it just be monolayer. You could have your proteins just kind of attached to another lipid. So you can, just like you could glycosylate, okay? You could do something like this with glycosylation and then stick stuff onto that. Or you can just have a protein attached on the surface. It's fine, it doesn't really matter. All of these things happen. One of the most important concepts to think about, though, is if you're going to have a transmembrane protein like these are, so there's like a beta-baroporin, which we're going to talk about in a second. When you're going to embed a protein, think about what you're embedding it into. Well, it's got charged head, hydrophobic tail, charged head, right? And so your proteins need to match that up based on the amino acids that they have. Okay, and so if I'm going to embed, like, for example, like this porin, the outer edges here, or the chunks of the protein that actually stick out, need to be hydrophilic. Sorry, I almost missed that. They need to be hydrophilic because the charged heads are charged. So you're going to want like your charged amino acids on those edges, whereas you want non-polar amino acids in your protein in the internal part. And that makes sense. Like, it's one of those kind of like duh things. But honestly, that's a really important concept. And that's why if you haven't taken biochemistry, that's okay. But if you have, it kind of makes sense. That's why we learn our amino acids, so that we understand like there are polar amino acids and there are non-polar amino acids. There are charged amino acids. And changing where your amino acids are located changes their functionality and can really have a huge impact on like where they're embedded. Are they even able to be embedded properly into like a vesicle wall or something? So the movement of something across a membrane, this should feel pretty familiar. We talk about these kinds of things in like a microbiology class. I really like this chart. I apologize, the quality isn't as high as I typically like, but I like this one because it's just a nice, clean way of showing this. So membrane transport is incredibly important. Membranes in general are incredibly important. 10% of the human genome is transport related. Okay, so 10% of all the genes in your body code for some kind of protein that's associated with transport through a membrane. That's how important it is, which sounds really cool. That's one of those fun facts where you're like, I mean, cells have to eat and talk to each other, so like of course it's going to be really high. Anyway, so diffusion. So we talk about the membrane is semi-permeable, right? So just simple diffusion, I mean, that's your gases and your water molecules. There actually are a lot of molecules, like glucose being one of them, that can have simple diffusion from high to low gradient through cell membranes. But in general, you want to have a large influx. So yes, glucose can pass through the phospholipid bilayer, though you will find places that say that it can't. There's a small amount of simple diffusion. However, you generally want like 1,000 or 10,000 or 1,000,000 times more glucose to actually enter the cell. And so that's where you facilitate that diffusion. And so as long as your passive diffusion is your high to low gradient, okay? So if you got a bunch of stuff on the outside that you want inside, or you have a bunch of stuff on the inside that you want outside, then you can just, take advantage of that. And that's your passive diffusion. That can be facilitated through a couple different ways. There's channels is generally what we think about. Channels can just be open. We call those pores. So there is a lot of vocabulary in here that gets used in the wrong place, or they'll use the things when they're synonyms when they're actually technically different. So you'll hear me kind of gloss over a few of those things. But basically, your channels or your pores, those are just like little holes, but they can be very specific. And the specificity is probably the most important thing to talk about with transport or transporters. Transporters being a more generic term for everything that allows something through a membrane. They're almost always proteins, though they can have other things attached, but it's just a protein. And for the most part, I don't know, they're just transmembrane proteins that let stuff in or out. I don't know, it's not that complicated. But I don't know, you can have a pore, okay, to let stuff in. Aquaporins would be for water. There are glucose transporters that are just straight up channels. But they can also be gated, which is really, really important. We're going to talk a little bit more about gated channels in a second. But that's, it's not always open, basically. There are also carriers, and carriers require some kind of energy that doesn't have to be ATP. Okay, so pumps would be carriers that use ATP, as opposed to co-transporters, which can take advantage of like the idea of alternating gradients. Vesicular transport would be the third way that we actually transport things in membranes. So that's like exocytosis and endocytosis. We are not covering that today. I'm moving that to actually near the end of the course, where we're going to talk about the cytoskeleton, and I lump membrane transport into there, like exocytosis, because they kind of fit really well together, because you have to understand the cytoskeleton really well to understand how endomembranes work. So talking about channels just a little bit, they're very selective. So they're really specific for whatever substrate they're trying to move. So remember, we classify these proteins as enzymes, and because they're lowering the activation energy for something, okay? They're allowing something to happen. Now, if that means it's opening up and letting something through, awesome. And so what it's actually allowing through, we call its substrate. And you can have a ligand-dependent substrate. So that would be like, I need something to bind to it to actually open the channel and allow something in. So we see that with like neurotransmitters and stuff. But there are other examples too. There are sodium-potassium channels that move. There are, when I say voltage dependent, so that would be requiring an electrochemical gradient, which that just means you got a bunch of positive charges on one side and a bunch of negative charges on the other side, or an overall positive versus negative charge. So like in a neuron along the axon. And so you can take advantage of that because it's a gradient, right? And so you can take advantage. If I have a bunch of positive charge on one side and a bunch of negative charge on the other, it's going to be a lot easier to move something from the positive side to the negative side if it's like positively charged. The other thing you can have, I had trouble finding temperature dependent channels, like an example of a temperature dependent. It seemed more like in creatures like bacteria even, or like single-celled creatures that would encounter different temperatures for the most part. We maintain our bodies at a single temperature, so it's not as common. But you can imagine, like, if you're a single-celled organism and you're swimming along and you go into like some cold water, maybe you need to change how you actually, like what kind of things you let into certain organelles and other things. So yeah, that's one example. And then the mechanical one, the one that I found was the ear cochlea. So you can imagine the movement of air as it hits your eardrum. That's, a mechanical shift that's causing a change, and it causes mechanical gated channels to actually shift. So that's kind of fun. But that's channels. And so in general, they're allowing something from a high concentration to a low concentration. Okay. Now carriers are a little bit different. They're not generally channels. The difference between a channel and a carrier is that only one side of a carrier is ever open, okay? So we call that alternating access. And so you just have alternating access. And so it's like a little thing that can shift. So it enters one side. So imagine if your sodium entered here, and then this side opens and it kind of like, it's like a little, like a teeter-totter almost, and it opens up. And so you only have access to one side. And so that's your carriers. Now, your carriers can still be facilitated diffusion. So if you're going from high to low, you can still use a carrier just like you would use a channel. It's just it's not always open, or it's just because. So let's imagine that this is a carrier. You got a ton of sodium on one side, and when a single sodium binds in, this can kind of flip open and let the sodium back out, and then it reverses, okay? Just like a little teeter-totter, opening and closing. It doesn't really care, like if there's a high concentration of sodium on one side and a low on the other, then yeah, it's gonna go that direction. So you can have facilitated diffusion as the power, because when we have a bunch of sodium on one side and a bunch of sodium not on the other side, that's a gradient, right? And that's our form of energy that we're using to power this. Now, we usually think about carriers as being going against a gradient. And that's where you can get into a little more complicated things, and we would call that active transport. Primary active transport uses ATP. So that's basically how we define that. Secondary transport is also called co-transport, and that's more like if you were going against a gradient, and instead of using ATP, you're using electrochemical potential. If you generated an electrochemical potential, just like I was talking about with like a neuron, if you generate an electrochemical potential, you can use that to your advantage because you have a large charge on one side and a different charge on the other, and you can take advantage of that. And you can do different kinds of co-transport that way. So these are kind of lumped into three categories. So there's uniport, which is just like one thing moving through, which imagine, if you will, if you had a bunch of positive charge overall on one side and negative charge on the other. And so the calcium ions would want to move from the positive to the negative charge. And so you can take advantage of that. And so there are active transporters that would take advantage of that, that allow calcium to, I guess, come either against or with a gradient. Okay, it just kind of depends. Symport would be two things coming in at the same time. So there are symport sodium glucose pumps that take advantage of, some of these are primary and some are secondary. Oh my gosh, I apologize. I get confused, to be honest, because you'll hear different terms used, and then I knew this was going to happen, and then you get tripped over your words, and I'm like, okay, like what can I actually tell you guys that's important? Well, two things can come in at the same time. You can send two things in opposite directions. There's sodium hydrogen pumps. You can bring just one thing in. How you physically use, what power you use to do uniport, symport, or antiport changes. So it could be either primary uses ATP or secondary uses something else, in general electrochemical transport. Okay, and so there you go. That's like my terrible way of trying to explain this. Honestly, does it really matter? If you're going to join a lab that's doing this kind of work, then you need to be, like maybe more detailed about what your vocabulary is. But there's active transport, which means you're going against a gradient. There's passive transport, which means you're not. That means you're going with the gradient. And you can have things go the same direction or opposite directions. It doesn't really matter. Like it just depends on what you're doing. So I have another slide from Draw to Know It that kind of talks about this. So sodium potassium pumps is probably the most common one that we hear about. That is an ATP dependent pump. You can couple things together as well. So at the bottom here, we have our coupled ones. Now, what's interesting is your coupled transporters, they might not directly require ATP, but generating an electrochemical gradient with ATP via like a sodium potassium pump could drive one of your other transporters. Okay, and so here are some examples, right? So sodium glucose. What is this? Just having sodium, like having a massive influx of sodium. So that would like trigger your neuronal, not synapse, but to trigger the electrochemical swap that happens along the axon. And then like having in your cardiac muscles, you have an antiporter for sodium and calcium. And calcium is really, really important for your muscles, and having your muscles actually trigger their contraction. And so having those antiporters is really important. And this is why you can start to see there's some connections to physiology here, right? So the amount of sodium and potassium and calcium that's in your diet is really, really important. You can mess that up. And so that's why, like in medicine, sometimes we measure like how much calcium do you actually have in your diet or in your bloodstream? How much sodium is really, really important. So the last thing I wanted to talk about is something that you guys are going to talk about actually in class, which are cell junctions. And so I lump this together with cell membranes because all of these junctions are integrally associated, integrated with our cell membranes. And so we need to understand, like the basics of a membrane if you're going to understand how to embed these proteins and thus use them to kind of look at a bigger picture, right? We're zooming out a little bit. So instead of just a cell membrane with proteins embedded, now we're kind of looking at that extracellular matrix and the paracellular pathways. So that would be like the pathways between cells. And how tight do those need to be, or how loose do they need to be? And that kind of changes based on your junctions. And so what I have actually in the slide set here, right here I have talking about all all five-ish of the junctions that I wanted to talk about. And then I have pictures of each of those on subsequent slides. Most of them are from Wikipedia. But so tight junctions, they're super tight. They prevent things from going through. And they can also prevent lateral transcellular transport. So that's just like the endocytosis or exocytosis between cells that are next to each other. And so that's something that you might not think about, but what it's doing is it's promoting the movement of things from one cell surface. So imagine like your intestines, right? So you've got your intestinal cell wall, and they have those little dangly things sticking out into the lumen of your intestine, and they want to pull nutrients in and towards the body. And so we have a lot of tight junctions along there between the cells, because those tight junctions are so tight, so you can see them here, they actually prevent, along that paracellular passage here, they prevent any exocytosis or endocytosis, I guess, exocytosis from the central cell into these other cells, which would then be endocytosis. So they prevent that. So they're just making sure that nutrients go from the inside the intestinal lumen into the body, okay? And so that's a function of tight junctions. And they're really, really tight. They're not perfectly tight. They can allow leakage depending on like what you need to have happen. But for the most part, they're incredibly tight, like watertight. Gap junctions are your most basic junction of like, they're just holes through cells so they can talk to each other. And it allows organs and organelles to function together. So that kind of makes sense. So if I have like, let's say your liver, and all those liver cells have gap junctions between them. So if I stimulate one of those cells, whatever I stimulate that cell with can sort of transfer to the rest of the cells as long as there's a gap junction. And so you can get what we would call bystander effects. We see that often with like cell death. So you can trigger, you could have like, let's say 100 cells stuck together, and I trigger death in one of those cells. Well, the death trigger can pass, like the cytochromes and things that are affecting the mitochondria to trigger apoptosis can pass through gap junctions. And it's a faster process than if I had to, send like a death factor to every single cell. And so that's just a bystander effect. But generally, it just lets stuff, it's a connection between the cytoplasm. They're just sharing cytoplasm. It can be pretty specific, or it can be pretty generic what it allows through. Adherens, all of these are associated with communication, which that's one of those just gloss over. Like every time you look up these things, it'll say like, it's associated with communication and structure. And you're like, well, yeah, but like, what does it actually do? Well, in general, adherens they connect to the cytoskeleton, and they just allow, that would maintain your structure. So you'd have your gap junctions to allow talk between the cytoplasm, but then you would have your adherens junctions to really make sure that the cells all kind of move together, which makes more sense like with your epithelials and like your organs, when you want your organs to kind of like stay stuck together. And desmosomes and hemidesmosomes kind of fall into those categories as well. So desmosomes are super, super strong. So we'll see those, they're kind of like adherens, but they're more for like mechanical stress. So you see them in cells that undergo a ton of mechanical stress. So like muscle cells and cardiac cells, cardiac muscle cells that are like constantly like moving. And so they just provide some nice relief of mechanical stress, or reduction in the damage that can occur from mechanical stress. And then hemidesmosomes are called hemi because they're on one side. So they don't connect two cells. They connect the bottom of like an epithelial cell to the basement membrane that it's attached to. And so that's generally where we see them, is on your epithelials, and I think your endothelials as well, where you don't necessarily want them touching another cell. You want them attached to like a base plate, and then they can like go from there. And so they're called desmosomes because they kind of looked like that, but they're actually relatively different. I have a nice picture. So I have pictures of all these. So there's like your tight junctions with the different proteins that are associated. There's your gap junctions. It's not listing the proteins, but you can look those up. There's your adherens, and you can see with the actin. And there's your desmosome. Okay, right? So super structurally sound, also connecting to the cytoskeleton. I mean, of course, but what you're going to do. And then you've got your hemidesmosome. Now, Wikipedia didn't have a picture of it. But so you can see, so up here, this would be like your epithelial. I mean, they're saying it's a keratinocyte here. But so you got your epithelial cell with some hemidesmosomes, or some desmosomes holding them together. And then you've got a hemidesmosome attaching it to that extracellular matrix. And that's really their function. So it really keeps them nice and holding tight. to whatever they're attached to. But you can see all of these things have, all these cell junctions have tons of proteins associated with them. Some are the same, but most of them vary based on what they are, and they all have different purposes. And just to remind you, think about all of these. So this one's got what, like 20 different proteins that are associated? Every single one of those, there's multiple copies of it. Those all came from a messenger RNA, a separate messenger RNA that maybe was spliced, and that all came from different genes. And all of those have to come together to be associated with this one spot on a cell. So they all had to come together through like the endoplasmic reticulum to be embedded. Some are embedded, some are not. Man, things get complicated in the cell, but that's why we're in this class, to learn that we don't know anything.